So, as Cora said, I'm a medical oncologist, which means that my job is to give chemotherapy to people. And if you've ever had the misfortune of having to sit on the wrong side of a medical oncologist, you'll know that most of the time when we look at a person, we truly do not know what chemotherapy is going to work best for them. And that leads to a lot of ineffective and very toxic treatments, and obviously something that we are very motivated to try to address. So we know that when you look at a person, we cannot actually guess at the kind of chemotherapy. We may be able to use technology to look behind these tumor cells and try to understand what's driving them. And so these were our motivating questions. And the last 2 decades of targeted therapy has given us a lot of hope. But the truth is there are still targets only identified in the minority of cancers. And I think the easy targets have already been found. And now the challenge is, how do we find all of the other niches and vulnerabilities in cancer? And we do know that cancer is really a disease of the genome, dysregulated growth driven by genomics and the environment. So this is what launched our project 12 years ago. We started in a project we call Personalized Oncogenomics, or POG, because we like to say we POG things, and we've used it like a verb and a noun. So we've coined a new phrase. So we started in 2012, and we take patients directly from the province. It's a very huge geographical area, but about 6.5 million people, and we take patients primarily with advanced diseases, and we consent them, we biopsy them, and then we do whole genome transcriptome analysis. We have a big platform of data sharing, which I'll show you later. And then every single case that sequence is discussed in a multidisciplinary tumor conference, which we launched to address these problems and to really collaborate and explain to the scientists and clinicians all of the different aspects of the patient and their cancer and the treatment options that we might be able to identify. There's a lot of spin-offs from this, hereditary cancer, clinical action may or may not be taken. We have a huge clinical database, and so on. And so we've been doing this now across the province. We have now enrolled about 1,400 people. The clinical data is something that I'm very focused in, obviously. You'll see at the bottom there's a couple of references for some of our major papers. And what I'm particularly interested in is that if you look across all different tumor types, because we enroll a wide spectrum of different tumor types, it's not just one type of data that drives our clinical actionability statements, that you can't just rely on the DNA or the RNA, but it's actually multiple data types that leads us to a decision about clinical actionability. And clinical actionability is something that we come to in the tumor group, where we say, here's the data, what could we truly act on clinically? And so this is always a good debate, as the genome scientists might present something and then we say, well, that's may or may not be clinically actionable. But a clinical actionability rate of over 80% is actually truly remarkable, particularly for patients where a lot of them have been heavily pretreated. We're not able to give this treatment to everyone. We are able to treat about 40% of people, which I still think is pretty good. And that might come up later in terms of why we're not able to treat more. But we're hopefully moving in that direction. So the question for this talk is really, we're already doing this short read sequencing. We've been doing it for a decade. We're really good at it. We all understand it, sort of, now. Why would we do long read sequencing, and what might it add? And how do we select patients? So that's the subject of this cohort analysis, where we actually decided to select about 189 patient tumors with 41 matched normal. And the reference below is Kieran O'Neill's paper that's just come out online, so you can look at it. What you can see across the top of this diagram is that this is multiple different tumor types, I think 25 different tumor types, but also from multiple different biopsy sites. So this is messy data, but that's actually what we have in the clinic. Patients are not homogeneous. These are messy, and they're heavily pretreated, and it's complicated. So if you can actually get usable data out of this more real-life experience, I think that's very valuable. So I'm just going to highlight really the methylation aspect of the data and how we as clinicians have come to actually think and talk about methylation, which is not something that we historically did. So one of the main things is that POG patients often are poorly differentiated cancers. We don't necessarily know where the cancer started from, and it's very helpful if we know where a cancer started from so that we can access treatments. And so usually we use RNA data to try to align a tumor type with a place of origin. We've actually found that methylation is really interesting and actually clusters extremely well. So we can build up our own libraries and compare. And and you can see that they cluster beautifully by these colors here. And then, this, for example, is a group of pancreas patients. But what was interesting is we found this little group of outliers, and we said, well, who are those pancreas patients that are not actually clustering properly? Actually, those were pancreatic neuroendocrine cancers, which we know clinically are actually quite different. So this was a really nice reality check to know that we are, in fact, be able to truly better differentiate based on methylation. And then we asked as a clinical team, well, are you sure you're not just actually clustering based on where the tumor was biopsied from? So are you getting all the liver biopsies together and not the different cancer types? And actually this again was a really nice check because in fact this is methylation based on where the biopsy was taken from, and it really doesn't cluster well at all. So this is really interesting for us, and we're now incorporating long-read sequencing when we don't actually know, when we have a tumor of unknown primary or we're debating where a tumor came from. And the second and potentially clinically really relevant is the data on HRD. So HRD, we know, can be measured by patterns of genomic changes, and it's really important for cancers like breast and ovarian because we think about treatments with platinum or PARP inhibitors. And so we know with breast and ovarian, for example, that you're really directly picking treatments if you can identify an HRD signature. But a lot of the times when we find an HRD signature, they're not able to actually say why. And this is a problem because if you can't tell me why someone has an HRD signature, then I'm not sure I believe it. So this was a study of this cohort of about 50 breast and ovarian cancers where we examined 51 HRD genes. And this is the data on the BRCA1 and RAD51C. And you can see the HRD detect score is on the Y vertical axis, and the methylation is on the x-axis, yes or no. So let's just look at the BRCA1 for a second. There are a whole bunch of cases where there's high methylation, high HRD score, but not methylated. And this is where actually all of the either germline or somatic BRCA mutations were, or RAD51. You can see that. I think I have a little arrow. There we go. So these were totally explainable. They have an high HRD. Oh, they're BRCA. Oh, good. Let's give them a PARP inhibitor. But then there were 6 cases, but actually now we've added to this data. So we in fact have 12 cases where there's a very high HRD score, but no explanation, no mutation. And as clinicians, we might say, well, I don't know that I believe that. You can tell me that, but I'm not sure I'm going to give someone a treatment based on data where you can't explain it. But actually, it could be explained by methylation. So if these genes were heavily methylated, then we have an explanation and potentially then therapeutic relevance. So this is just 3 cases you can see on the little diagram there. POG 804, 507, etc. Those are 3 different cases with the HRD score again on the little y-axis. And what you can see is the CPG islands, so where transcription starts. So let's just start with the light blue little triangles. That's germline, so that's properly unmethylated, so you can have proper transcriptome. And then you've got the other little circles on that lower line there are the normal, either the normal DNA or the normal tissue from the cancer sample. But that big purple line up at the top there is heavily methylated BRCA1. And so not only are we able to explain it when they show it in a graph, but they can actually like functionally explain it to me with, and that as a clinician, then I say, okay, I can take that back to a patient. I can explain it. I can say your gene is shut off. It's not mutated, but it's shut off. And therefore, we believe in this data and can potentially think about treatments. So this is the kind of way we're using this data, long-read sequencing, this cohort of 189 cancers. We're now prospectively looking at new collaborations, new ways to roll this forward, data sharing platforms, and so on. And prospectively now in our tumor boards, when we see someone, or even before, when we see someone with a high HRD but we can't explain it, or if we're not sure where the cancer came from or so on, we're able to select these patients preferentially for long-read sequencing. And I think that will probably expand as we go along. As I said, the data is readily available on not just the long-read cohort, but everything. So I would encourage you to go look at that if you're interested. These are my acknowledgements. And there's, of course, a huge team of people behind this. And actually, this is the whole team of people who participate in POG. This is what it takes to run a project like this. Thank you very much.